Right now, at this moment, you have cells inside you that are dying. Are you going to go and see a doctor? Are you going to go to the emergency room? Is this a bad thing? Not always. And that is why we have to talk about apoptosis. So what is apoptosis? Apoptosis is programmed cell death. And we are going to talk about exactly what that means in this video. So cell death, that's pretty understandable. But why is it called programmed cell death? Well, the reason why is because this is your body's planned out, calculated way of killing cells that it does not want around. So for example, we are constantly having certain cell types like red blood cells and skin cells just end their lives because they don't live forever. Some cells like neurons are more long-lived, but other cells that we call epithelial cells, which basically means the lining of anything, they die pretty quick. Also red blood cells, like I said, and maybe sometimes endothelial or inside lining cells as well, might die off. And that's a good thing. And why that is going to come up later in this video. But before we continue with apoptosis and why it is so awesome for us, let's compare apoptosis with another form of cell death that you might know, or you don't know that you know, called necrosis. So apoptosis versus necrosis. Apoptosis, the word actually came from a Greek poem about leaves falling off a tree. So leaves fall off a tree when they're ready, it's purposeful, it's meant to be, and it's to benefit the greater organism. That is the difference between apoptosis and necrosis. Necrosis is not planned. Necrosis is messy as heck. Necrosis will happen if you have what's called ischemia, which means lack of oxygen. Necrosis can happen just anytime you have trauma or injury. Necrosis is if you picture a wound where you think cells are dying and it's all like black and yucky. That's not apoptosis, that is necrosis. So the differences between them are outlined in this table, and you do not have to memorize this table, not at all. But to get the general idea that apoptosis is planned out, it's very, very neat. It's going to happen in a way that does not destroy the surrounding cells. So for example, in necrosis, when your cell dies, you secrete kind of all of these toxic compounds. And that's actually how you know when you're in pain, is because cells die unexpectedly, and those chemicals that are released, those noxious stimuli, bind to nociceptors, which are our pain-recepting cells. But apoptosis will not do that. You did not feel pain when some of your old red blood cells died today. You did not feel pain when some of your skin cells said, I'm retiring, bye-bye, let new ones grow in. You didn't feel pain with that because there were no dangerous kind of chemicals released from that. You also have like very structured methods of cleaning up the dead cell. So for example, the cell will break down its compounds via autophagy. What is autophagy? Auto is self, phagy. Anytime you hear phage or phag in biology, it means to eat. So it is eating itself, digesting itself. So the lysosome will leak its hydrolytic enzymes. We have DNA enzymes that will cut the DNA into very methodically sliced 200 nucleotide fragments after and before every histone. So every nucleosome gets cut up. And if you don't know what a nucleosome is, I've got a video on DNA structure. But the point is that apoptosis is neat. Necrosis is messy. So for the rest of this video, when I talk about cell death, I'm not talking about necrosis. I'm talking about apoptosis. There are two main types of apoptosis, or two ways to get to the same point. All the apoptosis ends the same, and we talked about how that ends. And we'll talk more about how it ends in detailed steps. But there are two starting points. The one starting point comes from outside the cell. Something is going to bind to a receptor, and this is how a lot of signaling pathways work, right? You have a receptor that spans the cell membrane, and when something, we call it a ligand, some people say ligand, both of them are correct. When this ligand binds, it's going to cause a conformational or shape change in the receptor, and the receptor is going to kind of relay this message through its conformational change to the intracellular components of the pathway. So in this form of apoptosis, we call this extrinsic. Extrinsic means it's coming from the outside. So some external ligand will bind to a receptor, and this receptor is called Fas. And Fas will change its shape and it will change its shape in such a way that it will be able to interact with a protein on the inside of the cell called FAD. What is FAD? FAD is fast-associated death domain. When FAS recruits FAD, and that's another thing that comes up often in signaling is recruitment. So basically when your protein changes shape, it allows another protein to bind, and because the cytosol is like a soup where all the proteins are floating around, it's gonna encounter that now active molecule and they can bind together, and that's called recruitment. So ligand binds to FAS, and the ligand is called FAS ligand. Very easy to remember that, right? And then FAS is going to bind FAD, fast-associated death domain, on the inside of the cell. And that is going to start a signal pathway that ends up activating these enzymes called caspases. And I like to think, and I think I got this from somebody, I don't think I made it up, that caspase is like Casper the Friendly Ghost. Because what are we doing? We are killing the cell. So these caspases are actually named this because they are C-aspartyl proteases. Aspartic acid is an amino acid, and C means the C terminus. So proteins have an N terminus, where the amino group on the backbone is just kind of hanging out, and they have a C terminus where the carboxylic acid group is kind of hanging out at the end. So C-aspartyl proteases are going to cleave at the carboxyl side of aspartic acid, and that's not something you need to memorize, but I like to know why things are named how they are. That helps me. And these caspases, there are many different ones. Some of them are going to go earlier in the pathway, and what they do is each caspase cleaves the next one. You might have heard of pathways where each protein phosphorylates the next one, and that's kind of their way of activating each other. For example, the MAP kinase pathway. In this pathway with the caspases, one caspase will cleave another caspase, that one becomes active, it'll cleave another caspase, and it will go on towards the end, where basically the signal gets amplified and everything that's involved, all the cellular machinery involved in apoptosis, will become active thanks to the caspases. So that's extrinsic. We started on the outside of the cell. Now we have intrinsic pathway, and the intrinsic pathway is starting inside the cell, to be specific, inside the mitochondrion. If we are going to destroy something from the inside, we're going to blow it up from the powerhouse, right? Our mitochondrion is our powerhouse. And inside the mitochondrion, we have this process going on always when the cell is alive called cellular respiration. And in cellular respiration, we have this thing called the electron transport chain. And if you don't know what that is, I have videos on that too, and you should know what that is. So in the electron transport chain, we have proteins that are embedded in the inner membrane of the mitochondrion, right? Complex 1, complex 2, complex 3. Nobody was getting creative naming those. And then we have some mobile electron carriers. So we have cytochrome C as one of those. And cytochrome C just goes back and forth between proteins and kind of shuttles electrons. And when it gives electrons away, it is going to give them to oxygen. And that's the end of the electron transport chain. Now, why am I telling you how the cell lives when I'm trying to tell you about how it dies? Well, because the intrinsic apoptotic pathway is going to start when pores form in the mitochondrial membranes. So these pores are going to let things in and out of the mitochondrion. Now, is that compatible with the cell's life? No, it is not, because if you remember from cell respiration, we had a concentration gradient where we had protons being pumped on one side of the membrane, and they all wanted to diffuse to the other side. If we make big giant perforations or holes in this mitochondrion, that's not going to be compatible with this process of diffusing the protons only through ATP synthase and only harnessing that energy to make ATP and not letting it dissipate, right? So we make these pores in the mitochondrion and the cell's life is kind of effectively over. But the other important thing is when we make these mitochondrial pores, we are letting out cytochrome C. That's why I gave you the whole spiel about cytochrome C. So cytochrome C is going to diffuse out of the mitochondrion. This is not normal. This only happens during intrinsic apoptosis. And when this happens, cytochrome C is going to go and activate all those lovely caspases we talked about in the intrinsic pathway. So here these pathways are merging. And the caspases will go and activate everything in apoptosis. So Those are our two pathways. Now, why do we activate those? I know we said that the start of the extrinsic one is fast ligand, binds to fast receptor on the outside of the cell. The intrinsic one starts when cytochrome C leaks out through mitochondrial pores that are being made. But, how do those things get triggered? Well, on the outside of the cell, for the extrinsic pathway, this can get triggered, for example, by cytotoxic T cells. Cytotoxic T cells are in the immune system, and what they do is if your cell has cancer, and it has markers of cancer on the outside, if your cell is infected by a virus, and it has markers of viral infection on the outside, if your cell is not doing so well, things tend to spread. Cancer spreads, infection spreads. So cytotoxic T cell says, I don't want that to happen. You are going to die before you spread this to anybody else in my precious body. So cytotoxic T cell will come with its fast ligand attached to it, and it's going to kill this cell. The intrinsic pathway, on the other hand, is activated by things inside the cell. So DNA damage, or just anything in the cell that is going on that is not supposed to be going on, that gives the cell a red flag, like, hey, maybe you're cancerous, then you're going to do intrinsic apoptosis. Something is going to tell the proteins that make the mitochondrial pores you need to be turned on. So now that we've discussed a little bit about how these pathways work, I want to talk a bit more about how they are used. So apoptosis has a few main uses. We talked about killing infected cells. Another one is development. So when we are a developing embryo, we make a little bit too much sometimes, and then apoptosis is going to help us to scale back where we need to scale back. So, for example, if we didn't have apoptosis, we would have webbed hands and feet, because you couldn't have the cells that are in your webbing as an embryo kind of recede via apoptosis. Apoptosis is also really important in cancer, not in making cancer, but in stopping cancer in your body. Because what is cancer? Cancer is a bunch of cells that divide aberrantly, aka they divide too much for their own good. They're going to divide really fast, make this really dense cell mass called a tumor, and that tumor can break off and metastasize and invade other parts of your body. So this is a really harmful process, and we want to stop it. How does cancer happen? Cancer happens via mutations in DNA. So genes make proteins, right? Central dogma of biology. DNA makes RNA, RNA makes protein. So your genes, if your genes get mutated, and they are coding for proteins that are involved in the cell cycle, like cycle-independent kinases, if they are involved in DNA replication, if they're involved in any of that, then if you mutate them, and mutations do happen because you have literally trillions of cells and they're all dividing at some point, mutations are going to happen, then what's going to happen is the cell cycle will get messed up, and somehow you will have too much division. And if your cell doesn't have a way to check that and stop that, that is how you get cancer. So apoptosis is supposed to stop cancer cells, and when we detect internal signals like DNA damage, etc., then the intrinsic apoptosis pathway is supposed to kick in. The ways that mutation can happen in cancer, and this is a bit off from apoptosis, but the two are so linked that I think this video is going to be kind of a combo, apoptosis and cancer, is there are two types of genes involved. One of them is called proto-oncogenes. So I'm going to write that out. Proto-oncogenes. And what does proto mean? Well, proto means before. What does onco mean? Onco usually means something to do with cancer, and a gene is a gene. So these are genes that, if they get messed up, they may cause cancer. But proto-oncogene is the term for them when they're normal, before anything has happened to them. If something happens, if there is a mutation, these proto-oncogenes will become oncogenes. So I said there are two types. This is the first type. Both of these, proto-oncogenes and oncogenes, are category number 1. And category number 1, proto-oncogenes, they don't have to be number 1. That's just the order I'm doing it. But proto-oncogenes that turn into oncogenes, they are originally genes that tell the cell, yes, you can divide. Do it. Divide. And they are sometimes on, and they're sometimes supposed to be able to be turned off. Your cell is constantly in a battle. Divide, don't divide, divide, don't divide. And you need the right number of regulatory signals in each direction. So proto-oncogenes say, yes, you should divide, but I can be turned off if I want to be. Oncogenes have a mutation so that it's always, yes, you should divide. You're trying to turn me off? No, you're not allowed to. We're going to divide no matter what you say. So oncogenes are dangerous because they just make signaling pathways go always on. They can never be turned off. And apoptosis is supposed to see this and stop this, and it does not always. The other type of gene is called a tumor suppressor gene. And this one hopefully is a little bit self-explanatory. Tumors are cancers, right? Suppressing means that we're going to turn it off. So while all the proto-oncogenes are trying to say, yes, cell, you can divide, tumor suppressor genes say, hey, chill, not right now. We're going to wait a little bit before we divide. And that's usually what they say. So if tumor suppressor genes are going to get messed up to cause cancer, do you think they're messed up so that they're always on or always off? Well, if you mess them up and they're always on, then they're always telling the cell, hey, chill, wait, don't divide. And that would lead to low division, right? But cancer is an excessive division. So tumor suppressor genes get messed up when they get destroyed, when they have a loss of function. So proto-oncogenes made into oncogenes, they were made into oncogenes by a gain of function mutation. So basically they had a function and now they always have a function. Tumor suppressor genes are made into a danger for cancer if they have a loss of function mutation. So for example, P53 is a famous tumor suppressor gene, and here we can see that it's supposed to sense DNA damage and say, nope, don't divide. In the bottom figure, it does not. So you might not have to know this if you're in an intro-level bio, but if you're higher up, if you're studying cancer bio explicitly, then you might want to know that tumor suppressor genes, you need 2 bad copies in order for your cell to start acting weird, right? Because if you have one of them that loses its function, the other one's still going to work just fine. If you have proto-oncogenes, you only need one to be messed up for your cell to be messed up, because if one of them is always on, then it's always on. Generally, loss of function mutations are going to be recessive. Gain of function mutations are going to be dominant. This is not just in cancer, this is in any gene, because remember, we have 2 copies of all our genes because we have a set of chromosomes from one parent and from the other parent. So apoptosis is supposed to stop all this crazy stuff. Sometimes it doesn't, and that's when cancer happens. Cancer actually happens when you have many times that your body fails apoptosis. People have come up with a theory called polyclonality, which basically means that any tumor that you pick up and analyze, it's going to be heterogeneous. It's going to have lots of different cells with different mutations. And that's what makes them so hard to kill. But when people try to kill them, they try to exploit the mechanisms of apoptosis and say, how can we use the death pathways that already exist in order to make these cells die? Or where are these cells messed up when they should be dying and they are not? So hopefully this helped clear up some misconceptions and taught you some cool things about apoptosis and why we need death in our lives.